Class 12th, this video will be explaining the process of RNA interference, the main concept, and its application to generate the plants resistant to nematodes, right? How that nematode infestation can be prevented in plants, right? First of all, some key words or key points that are supposed to be kept in mind. So here the first thing is the concept. Then you write RNA interference. That means your operative word. The definition is silencing of the target. Now target is operative here. Target messenger RNA. So when you are saying the silencing means you are not allowing this messenger RNA to express itself. You are not allowing it to complete its protein synthesis. So its expression is silenced or stopped. And when you say the target messenger RNA, that means you have already identified some specific genes which are nematode specific genes, right? Some specific genes of nematode, and you know that if you silence the expression of that gene, the nematode will not be able to survive. So what we do, we take some complementary, we take those genes which are going to work against, their expression is going to work against the nematode, right? So here 2-3 words that you have to keep in mind is, first of all, silencing of the messenger RNA, target messenger RNA, so that its expression in the form of protein synthesis or in the form of translation can be stopped, right? If suppose, let me erase this. If suppose you have to go through a flow chart so as to explain what are the main events here, so the main thing as the raw material that you have to you will be needing is the nematode specific genes. One thing, nematode specific genes, right? This is your first keyword that will be giving you the points, and you will be using these genes in the form of double stranded RNA. Now double stranded RNA can be made with the help of RNA viruses. These are retroviruses that can perform reverse transcription, or maybe the jumping genes which are known as transposons. These are mobile genes, they can move from one place to another, and allowing these to equipping them with a reverse transcriptase virus, so that a reverse transcriptase enzyme, so that if suppose you have one strand with you, the other strand can be generated. This is one RNA strand, the other strand can be generated by reverse transcription. Reverse transcription, right? This is how both the strands are there, but both are RNA strands, so you call them double stranded RNA. Okay, so this will give you 1 point, one tick. And thereafter what you have to mention is that you will be dealing with 2 locations here. One is the tobacco leaf, because the nematode infestation is much evident in tobacco plants, and the second location will be the body of the nematode when it will eat the tobacco leaf, right? So in the form of a flowchart, if you will begin with tobacco leaf. I am giving you a flowchart form, right? Tobacco leaf, then insertion of double stranded RNA in tobacco leaf, right? Once this double stranded RNA, which is specific, I am writing here specific because these RNAs contain nematode specific genes, right? When these doubles and you will represent it this way always, and when this double stranded RNA is introduced into the tobacco leaf, remember, I did not write it here. Remember that this is done with the help of agrobacterium. because you know that this bacterium is a tumor inducing bacterium. It can attack, it can incorporate the genetic material directly into the target cell without taking help of any intermediate, right? So agrobacterium will be directly introducing your double stranded RNA into the tobacco leaf. Once the double stranded RNA is entering the tobacco leaf, then the cytoplasm of the tobacco leaf cells contains the ribonuclease category, which is known as dicer. These are ribonucleases because your raw material is double stranded RNA. So ribonucleases are dicers, and as the name suggests, what they will be doing? They will be cutting these RNA, double stranded RNA, into siRNA. Now siRNA is small or short interfering RNAs. Interfering RNAs, right? So this is your raw material now, which you will be using further, because these small RNAs, you will be expressing them this way. These are still double stranded, but now they are small fragments, and they will be helping you to silence the target messenger RNA, right? Thereafter, just hold on, I need to create some space here. Once these siRNA are obtained, now they are double stranded, children. They are double stranded, right? Out of these, one is sense strand for you, and the next one is antisense. Only one will be having those uh, target silencing genes, but the other one is not actually needed here, because you need only one strand which will be going and getting combined with the target messenger RNA. So in that case, the next strand is supposed to be removed, and this is removed by the formation of RISC complex. Now RISC complex is the complex which is made up by the small interfering RNAs plus a ribonucleoprotein, which is known as argonuate. Argonuate. This is a ribonucleoprotein. When these two combine, this argonuate helps in removing one side of out of the two strands, removing one strand of this double stranded RNA, and only one is actually left, right? And this is your target RNA, which will be moving into the nematode's body, and in the nematode, wherever in the nucleus of nematode cell the messenger RNA is waiting for translation, it is waiting for translation, but before it gets translated, this single stranded RNA which you have processed till now will go, because it has the complementary sequences, it will go and get bound with this messenger RNA. So now messenger RNA is no more a single stranded RNA, it has a complementary bonding of the opposite strand. So its expression is actually stopped. Expression is actually stopped, right? This is how you are going to explain that how nematode infestation is stopped. This was a flow chart, children, but we can do one more thing. We can go with the diagrammatic expression as well. Just allow me some time. These were the keywords that I told you, how you have to proceed. Right. So suppose if you have to move diagrammatically, if you have to show this diagrammatically, straight away come to a tobacco leaf, right? This is a tobacco leaf. I am writing TL here, tobacco leaf, and now you are using an agrobacterium, right? This agrobacterium will be transferring your double stranded RNA, which contains, all these are keywords, children, which contains nematode specific genes, right? Thereafter, better to use a different color so that you don't get confused. Nematode specific genes. Now this double stranded RNA, right? There is a long strand. It will be acted upon by dicer. Now dicer are ribonuclease enzyme which will be cutting these long fragments into smaller one, right? This way. And now these are known as small or short interfering RNAs, right? But out of these two, one is needed by you, which is the sense strand. The other one is not needed by you because it is the antisense strand. I mean, the sense strand is the one which will be going further and stopping the translation of your target messenger RNA present inside the nematode, right? So what happens further? This small RNA, which is still, I mean, double stranded, though it is smaller in the length, but it is still double stranded. You need to remove one. What you have to do is you need to remove this one strand out of the double strands, and this is done by these two strands converted into one. I mean, one is only left, the sense one is left, and antisense is gone. This is due to the formation of RISC complex. Always write this way and then tell the whenever you explain, you write, you mention that here the siRNAs, small RNAs, they get combined with the ribonucleoprotein which is known as ARGONUATE, right? And this ribonucleoprotein actually helps in removing that antisense strand from the double stranded RNA, right? So ultimately, what are you left with? You are left with only one single strand, and this strand is going to stop the expression of messenger RNA inside the nematode body. Now let us take the body of the nematode, right? Suppose this one. So this is the body of the nematode, okay, and this single strand is now going in. What it will be targeting? It will be targeting the messenger RNA. Suppose we talk about, this is a nematode and we talk about, suppose one cell, whatever one cell is doing, all will be doing the same. So one cell is having one nucleus and one nucleus is having messenger RNA which is going to express itself. So if I say that this is the messenger RNA formed inside the nematode body and it is very much vital, you have to cancel it out. So the nucleotides present on this messenger RNA have a complementary bonding present on your RNA that you have introduced to this nematode. That's why you have used the word nematode specific genes, because the double stranded RNA you are making, it is complementary to the strand formed here. So it will get bonded very easily. So this kind of bonding between the siRNA, single stranded siRNA, and the messenger RNA of nematode, right? This will cancel out the expression or translation, any word you can use, translation or expression, this will be cancelled out, and this particular protein which was going to be synthesized will not be synthesized anymore. And this is how ultimately after some time the nematode dies because of the lacking of those vital proteins which did not form due to silencing of their messenger RNA. Right, children?